these extremely variable conditions of the variables that we were measuring was associated with rain events. And in our case, we defined a rain event as the amount of rainfall that fell within 2 days prior to our sampling date. And so the rain window began 12 a.m. 2 days prior and ended an hour before our sampling time. So that was the window in which we checked with NOAA and the Weather Underground records for how much rainfall fell during that time. Oh, I'm supposed to do this. So here is the Dutch Kills Basin, and it is an ideal site to determine the immediate effects in time and space of CSO activity. So these two pipes are what we call Tier 3 combined sewage outfalls, in that approximately 10% of the effluent that comes out of those pipes is untreated sewage. And the effluent is going into a part of the creek that's essentially a basin. As you can see, it's walled in. This would be the west shore, and this is the, I guess, the south shore. But in any case, this entire basin is pretty much bulkheaded, and it is scooped out so that the water really doesn't mix with the main waterway. And thus you can get a good idea of the effects of CSO outfall in the immediate sense. Okay, Enterococcus. Enterococcus is a bacterium that lives in the human gut, and CFU refers to colony-forming units. And Enterococcus is now the standard measure for the presence of untreated sewage. And colony-forming units per 100 mils is the common measure, and simply means the number of viable cells per 100 mils of water. And we're looking at this measure of density, bacterial density, in comparison with the rainfall in that window of that time period I just told you about. And we see what is essentially a direct correlation between the amounts, the concentrations of bacteria in the water, which is the red line, and the amount of rainfall in the government records in the blue. And what this tells us is that indeed the sewage in that water is not coming from some unidentified illegal source from local establishments. It's coming from those combined sewage outfall pipes, which are active during rainfall. Okay, so in the combined sewage outfall, sewage from businesses and residential toilets is mixed with street runoff. And when that street runoff is increasing in volume because of rainfall, it's diverted away from the water treatment plant and it enters the waterway. The dates at the bottom are our sampling dates. As you'll see, most of them are 7 days apart, but sometimes we sampled on Tuesdays and Thursdays. And of course, that window, that 2-day window moves with us, whatever the sampling date is. Now here we're looking at dissolved oxygen, and we're seeing the same profile, the same rain profile. So we're just missing the dates. The dates are the same. Now here are these precipitation maxima where we had the most rainfall, but now these are coinciding with crashes in dissolved oxygen, okay, the amount of oxygen that is dissolved in the water necessary for life. So now we have an indirect correlation between rainfall and available oxygen. Similarly, we see that when we have peak levels of oxygen, we've had very little rainfall in the preceding days. Now, also of interest is that there are crashes in oxygen, and here again, even when there was little rainfall. So what could account for this? Well, if we look back and see when the peak oxygen levels were, we find that the water contained at those times, and this is water super saturated with oxygen. This is our mean, the amount of dissolved oxygen that saturates water at 25 degrees Celsius about 75 degrees Fahrenheit, is about 7 milligrams per liter. So there are these wild fluctuations around the average. Now here at these supersaturation events, when there have been very little preceding rainfall, we see that the water contains an abundance of plant life. And plant life in the creek is at the microscopic level. So these are single-celled, or diatoms and dinoflagellates, single-celled plant-like cells that are photographed through the microscope at about 100-fold magnification. And we saw this type of phytoplankton density, okay, density of plant-like cells in the water, with each one of these peaks. Why did this happen? Well, let's get back to what causes these crashes in oxygen with precipitation. When that effluent comes out into the water, it's carrying organic material. Not only fecal matter from the toilets, but petroleum residues, oil from restaurants, these are all hydrocarbons, pesticides and fungicides and herbicides, these are organic material. So essentially we're providing a bountiful banquet to the bacterial populations in the creek, and of course the bacteria that are coming in with that fecal matter. Everybody's going to be decomposing this great food source. And to decompose these large hydrocarbons, proteins, nucleic acids, from huge molecules down to very simple ones, carbon and oxygen, carbon dioxide, nitrogen, oxygen, nitrates, phosphorus and oxygen, phosphates. To do that work, that decomposition, requires oxygen. So oxygen consumption, cellular respiration is powering the decomposition and providing all these nutrients as all this organic material decomposes. So now there's an abundance of fertilizer for those little cells. So we get a plankton bloom, an excessive amount of phytoplankton, so much oxygen is being produced by photosynthesis from these cells that is pouring out of the water. Now what happens when those nutrients are used up? Well, you have this huge biomass of plant life, and it's going to decompose. Another banquet for the bacterial population. And hence you have a secondary oxygen crash. So that CSO effluent sets up these wild oscillations in dissolved oxygen. And what are the effects on animal life of this? Quite amazing. Okay, so these two photos were taken at these two sampling dates, both very low oxygen levels. Now, warm water fish generally need a minimum to survive of 2 milligrams per liter of oxygen. So these are surface oxygen levels. Now we've fallen below that. We looked down from the bulkhead, so this picture was taken looking down into the water at our feet. And here we saw this amazing sight of an American eel, which is a nocturnal fish, active at night. During the day, it usually spends its time in hiding in crevices at the bottom. And here was this fish moving ever so slowly. And I don't know if you can tell, there's a little light there, but the mouth was breaking the water. The fish was gaping. And in very low oxygen conditions, fish can get just enough by turbulence created by swimming at the surface, which mixes the surface water with the air, just enough to survive. And he was moving out of there, but he had to move slowly. Not enough oxygen for rapid swimming. The next day, we pulled from our toe in the water, which Holly will mention, the plankton toe, where you pull a net through the water. We picked up this little organism. Anybody recognize those? It's a juvenile. It's a juvenile horseshoe crab, which is not a crustacean. They're actually more related to trilobites, ancient trilobites, than a blue crab that's also in the water. Now, again, it's not supposed to be here. This was actively swimming around. That's why it's not quite aligned with the grid, which shows millimeters, units. So this little guy was about 4 millimeters. And at that point, they settle on the bottom and burrow in the sand, because this is perfect food source for the little striped killifish, which you'll see in the back, which are abundant in the creek, and particularly in Dutch gills, they can survive low oxygen levels. They are unique in that regard, the extent to which they can survive low oxygen levels. Now, this abnormal behavior puts both of these animals at risk. As Fung and Willis can tell you from their observations of the American eel and the bird life in the creek, these are food for birds, just like these little guys are food for fish. So when they're forced to change their behavior as a result of these very low oxygen levels, their survivability is going to be diminished because they become easy prey for the predators. Okay, so we saw that oxygen levels vary greatly in time. They oscillate over time. They also are extremely variable with space if we look at depth. So here is our surface measures that we saw before, and here we are at 1 meter depth, and here we are at 2 meters. And you can see that at 2 meters in this environment, there are anoxic, oxygen-deprived conditions for much of the time. And what that means is there is no aerobic, oxygen-requiring life that can survive on the sediments in this part of the creek. And what should be a rich benthic environment is probably barren ooze sediment because of these low oxygen conditions. Why are they here? Well, those particulates that come in with the CSO, they are heavy, and they gradually settle out from the water over time and collect on the sediment, in the benthos at the bottom. And decomposition continues there until oxygen is used up. And when there's a little more oxygen, decomposition continues and then causes it to plunge again. And if there is sufficient time from the last rainfall event, eventually there's the sediment settles enough, or the particulates precipitate to a sufficient degree to allow enough light for photosynthesis for these algal cells to actually reach the bottom and provide oxygen. And they reach the bottom because now enough of the particulate matter has settled, enough light can reach the bottom, these cells can survive, provide oxygen, but this happens very rarely. So, extreme variability, and I need time. Quickly, pH is another one. With every peak rainfall, we get acidic conditions, and water acidity is damaging for the life in the water, just as low oxygen is. For example, in conditions like this, juvenile clams or mussels cannot develop a shell, build a calcium carbonate shell, at the time they need to settle from the water column onto the bottom. So acidic conditions are damaging to life, and let's skip that. So all of these factors, these poor water quality conditions with respect to dissolved oxygen fluctuations, fluctuating pH, high turbidity levels, they can be mitigated if the natural habitat were present, and that is the salt marsh. But the question is now, if you have a walled-in basin, and this is my last slide, how can we use the walls to bring back, to restore the natural habitat, which could then remediate the water quality of the creek? And why? Well, salt marsh contains, the healthy salt marsh, Jamaica Bay, for example, 10,000 mussels per square meter. And these are little filtering machines. An adult mussel can filter 50 liters of water a day, removing bacterial cells, removing phytoplankton. And indigestible sediment, they wrap in a mucus coat and deposit it anyway. Okay? Plants themselves can sequester heavy metals in their tissues and provide oxygen. So there you have ideas that have been floated around amongst members of this audience, and I will turn it over to Holly. Thank you.
I'm actually just not going to say very much real quickly because I know we need to keep moving. I just wanted to mention about our next steps for LaGuardia is that my students and Sarah's students will be continuing to collect water quality data. We're going to be adding another site, hopefully at English Kills, maybe. We'd like to add an even more polluted site, if we can get it all done. And we're going to be focusing, actually my student that's focusing on this is here, Carmen. We're going to be focusing more on plankton, and we even have our big nets back there to show people if they're interested. But we'll be focusing on the plankton that you saw on the one slide with the plankton bloom. We're going to be focusing on the abundance and the diversity of plankton that we see over time and over space within our sampling areas. So it'll be Dutch Kills, by the Nature Walk, and possibly English Kills. So we'll be looking more at the organisms as we continue on.